All right, so this is a practice AP question from your homework, and it's from an actual AP exam. And this sort of combines gene regulation with a little bit of genetic transformation. So let's take a look at this. So it says the regulatory sequences of the operon controlling arabinose metabolism, the ARA operon, were studied to determine whether bacteria can respond to changes in nutrient availability. So just to clarify what we've read so far, this is the operon. And these genes, B, A, and D, these structural genes, they code for making enzymes to digest the sugar arabinose. So if the bacteria eats arabinose, it will need to digest it. And so if you remember, a promoter, this is where polymerase would bind, and polymerase is what builds the mRNA. So the promoter is where polymerase binds. It would bind here, and it would go down this, and then it would make the mRNA, and then that mRNA would then code at the ribosome to make the enzymes to digest arabinose. But they're talking about whether they can respond to nutrient availability. In other words, the operator, this is the switch. So if they haven't eaten any arabinose, there's no reason for them to turn this operon on. I only need to digest my food if I ate something, right? So this is normally in the off position. This is blocked, typically by the regulator. If they eat arabinose, the sugar itself, will be the trigger that will remove the block, turn this on, and then it will make these enzymes to digest arabinose. So that's what this first paragraph explains. Then they say, we're going to do a genetic transformation. We are going to cut out these genes. We're going to replace them with the gene to make GFP, green fluorescent protein. This is a jellyfish gene. So now, if they eat arabinose, sugar, this will get turned on, but instead of copying these genes, it's going to copy this gene, and this gene is going to make the glowing protein, and the bacteria are going to glow. So they did this, they created this operon, but they put it in a plasmid. So remember, a plasmid is a circle of DNA that bacteria usually have, and it usually gives them some kind of a benefit. In this particular case, so this is the GFP gene, operon, we're sticking in here. So this is the operon. But somewhere else on this, if you read, it says that this plasmid also confers ampicillin resistance. So in other words, already on this plasmid, in addition to the GFP, there is also a little section here, and this gene makes them resistant to ampicillin, meaning if they're exposed to the antibiotic ampicillin, they won't be killed by it. So now they do a transformation experiment. So they take bacteria, they put them in a little vial, they add a bunch of copies of this plasmid, and then they probably put it through heat shock or whatever. And then some of them, remember, are going to pick this up and some aren't. So some are going to be positive and they actually have the plasmid and some don't. And now they grow them in 4 different media. So agar is just the food. Technically, it should be nutrient agar or something like that, but this is the culture media, meaning the stuff they're growing them on. In some of these, they add ampicillin, which is, remember, our antibiotic, and in some of these, they add arabinose. And then they have what color these colonies, the bacteria, are under white light, and what color they are under UV light. Now, just to clarify, GFP, if you read here, it only glows under UV light. All right? So before we even answer the questions, just let's walk through what's going to happen here. So if I have bacteria, and some of them have plasmid and some of them don't, right? So this is my sample. It's a mix of some that have it and some that don't. If I just grow them in food, then they're going to basically grow, right? There's nothing to kill them because there's no ampicillin added, so they're going to all live. And they're also not going to glow. Under UV light and under regular light, they're white. Now, why don't they glow? Because, remember, in order to turn the operon on to make the glowing protein, there has to be arabinose present. They explain that they're using an operon that already needed arabinose to turn it on, except they replaced these genes with the one to glow. So they have to have arabinose for it to glow. All right, plate number 2. I add food, I add ampicillin, but I don't add any arabinose. They're all white, and they're all white. That makes sense. However, if I add ampicillin, even though they're all going to be white and they're not going to glow, they will all be transformed. In other words, all the bacteria growing on this plate should be positive for the plasmid, because if they didn't pick up the plasmid, the antibiotic would have killed them. So on this plate, we have a mix of positive and negative bacteria, and we wouldn't be able to tell them apart. On this plate, even though they don't glow, we do know they all picked up the plasmid, because ampicillin would kill them if they didn't. Plate 3, we gave them food, which is good. No ampicillin, so nothing is getting killed. And then we add the arabinose. Now remember, the arabinose is the trigger to make them green. So under regular light, they're white. Notice under UV light, most are white, but some are green. Why aren't they all green? Well, because we didn't add any ampicillin. So we're not killing any of them. The ones that are positive are going to glow green because they have this operon that's going to make them green. The ones that are negative are just going to be white. So we're going to get a mix of green and white because everything's growing. And now in our last plate, we put our agar, our food. We add ampicillin, and we add arabinose. On this plate, they're white under regular light, but under UV light, they're all green. Why are they all green? Because they're all positive. Because the negative ones would have been killed by the ampicillin. So this plate, the reason they all can glow is because they all picked up the plasmid. So, quickly, the questions. If the normal bacteria has the operon, what happens when it's exposed to arabinose? Well, they explain that in the first paragraph. When you feed them arabinose, it removes the repressor, turns this on, and it makes the enzymes, it makes basically mRNA, which then makes the enzymes to digest the arabinose. Number 2, which plates have only transformed bacteria? That's going to be plate 2 and plate 4, because those are the 2 plates where we added ampicillin, which would have killed any bacteria that did not pick up our plasmid. So that would have screened them, so to speak. Why aren't they all green? I just explained that. On plate 3, we have some that picked up the plasmid and some that didn't. So we have a mix. And then what is the purpose of plate 1? Well, plate 1, we expect them all to grow. So this is a control group, but specifically, this is making sure that our agar is actually capable of growing bacteria. I'm not adding anything to kill them. We're basically making sure that we did indeed start with live bacteria at the beginning of this experiment. So you wouldn't just say a control group. You would say, well, this shows that we had live bacteria at the beginning of the experiment. It shows that our agar is capable of growing things. And so that's what you would want to say, not just control group. In fact, you might even say a positive control group. All right, so hopefully that helps to explain that question because I realized that it's kind of a difficult question, but you are fully expected to be able to answer questions like this on multiple choice or free response on your test and on the AP exam as well.